dear students, we are ready with the next lesson, next topic, and previously we had discussed about the restriction and the nucleases, and we came to know that there are two types of the cleavages produced by the restriction and the nucleases, as they are known as the symmetrical cleavages or the blunt ends. So there is an example of BAM-H1. This is a restriction and a nuclease which we obtain from Bacillus amyloliquefacient, and the site, that is the recognition site of this BAM-H1 is GGATCC 5' to 3' and 5' to 3' and the another enzyme is HAE2, which we obtain from Haemophilus ejectus. And the recognition sequence of Haemophilus ejectus is GGCC. So, can you now tell me if this recognition sequence was cleaved by BAM-H1 at these particular sites and Haemophilus ejectus recognizes this sequence and cleave at this site, what kind of the ends, cleavages it produces? So, what type of the cleavages it is? This cleavage is the staggered cleavage. And what type of the cleavage it is? It is the blunt and type of the cleavages. So, in the recombinant DNA technology, this particular restriction and nucleases have a very specific role. That is, when we have to isolate the gene of our interest, we have to recognize the restriction and nucleases which can cleave nearest to that particular sequence. And how they are used in criminology or forensics, and the application is Rflp. Very important. Restriction, fragment, length, polymorphism. What is this restriction, fragment, length, polymorphism? As the word indicates, restriction means we are using the restriction and nucleases here to cleave the segment of the DNA which in different individuals, individual A or individual B, which in different individuals when cleaved by the same restriction and nuclease, say it is EcoR1, and due to the individual differences we can get different fragments of different size fragments of DNA. That is due to the individual differences, and so the technique is termed as restriction, fragment, length, polymorphism. Means poly means many, morphism forms, means we are getting the fragments whose fragment length is of different sizes when they are cleaved with a particular restriction endonuclease. And this Rflp is used in forensics and also known as DNA fingerprinting. This is the basics of DNA fingerprinting. So, can you think over the concept? How we always say that DNA of every individual is different, and by this particular DNA we can identify the particular individual. So, DNA fingerprinting is due to the Rflp, or DNA fingerprinting and Rflp are same. Say, how it is used? We get a hair from a crime scene, which we know that it is of a criminal, or the semen from the body of the women who get assaulted. And from these sources, hair or semen, we get the DNA. We isolate the DNA from these cells, or the source may be the blood drops. If the crime scene is murder, and the first thing which we'll do is to isolate the DNA of any of the source which is present at the crime scene. And now there are a list of criminals. We have to isolate not criminals but the suspects. We think that there are 3 suspects, ABC, and who is the criminal? So what we will do, we will isolate the DNA from suspect A, suspect B and suspect C. So this is the DNA of suspect A, DNA of suspect B and DNA of suspect C. What we will do? We will isolate this DNA and fragment it all. All DNAs are fragmented with the same restriction endonucleases. And there are the individual differences between these 3 individuals, and the fragments which will receive are separated with a technique known as gel electrophoresis. And this will A, B, C, and we got the fragments, different size fragments. Now the question arises, what is this gel electrophoresis and how it works? So we will discuss it later on. And the resultant graph is compared with the graph which we obtained from the DNA isolated from the crime scene. And whenever it matches, we can compare. This is a simple principle. We will see it in the next diagram carefully. Means we have isolated the DNA, fragmented it with the help of the restriction endonuclease, and run it on the gel electrophoresis and compare with the DNA which we have isolated from the crime scene. This is the basic principle. This is the basic protocol. And how does this work? Now, firstly, we should come to know about the gel electrophoresis. What is this gel electrophoresis? This gel electrophoresis is a technique in which the DNA fragments are separated, and these DNA fragments are separated on the basis of their molecular weight. These DNA fragments are separated on the basis of their DNA molecular weight of the DNA. How? The smallest of the DNA fragment moves downwards. Smallest will move downwards while largest will at the top on the basis of the pore size. Smallest will run fastest, so it will go downwards, while the largest have the most difficulty in moving downwards. How does this happen? This is a gel which we prepare. We have holes which are known as wells in it, means the spaces, blank spaces in which we put our DNA content. We put the DNA content in it and run under the electric field. Negative charge here and positive charge here. This DNA, which is having the different fragments in it, now these different size fragments get moving downwards, and the heaviest molecule will remain at the top. The then lighter will move downwards, lighter downwards, and lightest at the bottom. So on the basis of the molecular weight, they get separated. Okay? And this DNA fragments of the genome are different in different individuals. So now move to the Rflp. What we have seen in the question, there is a sample from the crime scene, suspect A, suspect B and suspect C. DNA isolated from various samples are isolated, cleaved separately by same restriction endonuclease, and different fragments are put different. These are different, means from crime scene, sample A, sample B, sample C. Now move under the electric field. When it is moved under the electric field, the DNA fragments get separated on the basis of their sizes. The results may be any. And it is known to us that the DNA fragments which we obtain from various cleavages by different restriction endonuclease are different in different individuals except in the identical twins. So any of the living organism, any of the human being, any of the animal don't have the same fragments, produced same fragments, maybe same by the one restriction endonuclease but not by another restriction endonuclease, but identical twins have same results, so they are very tough to identify. And in this case, we can easily identify who is the criminal from 3 suspects. This is the DNA which we got from the crime scene, and this is the suspect, and there is similar banding patterns between this DNA fragments and this DNA fragments, and thus we can say that person C, the suspect C, is the criminal, and this is all about the DNA fingerprinting or Rflp. And the next is other enzymes which we have to use in recombinant DNA technology. We say that enzymes are the first tools, enzymes, and we have already discussed the restriction endonuclease, and the next enzyme which we have to discuss is the alkaline phosphatase, and another is the DNA ligase. These 3 are the major enzymes. ये हमने डिस्कस कर लिया है, and now we will discuss the alkaline phosphatase. Alkaline phosphatase is an enzyme which is responsible for removing the 5' phosphate group. Removing the 5' phosphate group. What is the function of this and how it acts? Actually, as we have studied that when DNA is cleaved with the help of EcoR1 or BAMH1, it produces this type of the cleavages, and the result is this. What is there? If this is 5' and if this is, then this is, and this is phosphate. There is a possibility of having the complementary base pairing between this and this due to GGATTC. Say in this. What happens when it cleaves? The resultant is this, and here the resultant is this. What will happen? If these are very near to each other, they can complementary bind to each other. How? These are binded, these are binded with the hydrogen bonds. This will move to its complementary, this will move to its complementary, it will move to this complementary and this. So what will happen? They will recombine. So there is no logic of separation or using the restriction nuclease. So whole of our procedure is disturbed. And why this happens? Because of the presence of phosphate group, which is present here at the 5' end and here at the 5' end. They will come together. Hydroxyl group is here and hydroxyl group is here. They can easily form the bond between them, and whole process combines together. So we have to use another enzyme that is known as alkaline phosphatase, because what will happen then? This alkaline phosphatase has potential to remove the 5' phosphate group. Means this phosphate group is now removed, and when a phosphate is removed, then here hydroxyl group is here. यहाँ पहले से ही हाइड्रोक्सिल ग्रुप था, यहाँ पर भी पहले से ही हाइड्रोक्सिल ग्रुप था, यहाँ पर फॉस्फेट ग्रुप था जो इस एल्कलाइन फॉस्फेटेज की वजह से अब कन्वर्ट हो गया हाइड्रोक्सिल ग्रुप में. एंड नाउ there is very less chance, even no chance of bonding between these two, because this cannot bond with this and this cannot bond with this. So we can separate these two strands. From where this alkaline phosphatase can be obtained? This alkaline phosphatase we can obtain from the calf intestine. If we obtain it from the calf intestine, then it is known as calf intestinal alkaline phosphatase. And when we obtain it from bacteria, we know the name is that bacterial alkaline phosphatase. So this is the second enzyme which is used in the RDT. First is restriction endonuclease, second is alkaline phosphatase, and now the third is DNA ligase. We have already studied in the about the DNA ligase or D ligase. Have you always heard this name? What is ligase? It is a ligating enzyme, means joining enzyme. And this DNA ligase we obtain generally from T4 bacteriophages. And this DNA ligase is responsible for forming the phosphodiester linkage between two adjacent DNA. Means say we have this plasmid, we have this gene of interest, these are the overhangs. Say this is the resultant of a staggered cut. So what will happen? They can easily bond. But as they do not have the alkaline phosphate group due to the alkaline phosphatase, here now the bond is formed with the help of this DNA ligase. Means it is used for ligation. So up till now we have discussed about various enzymes which are involved in the recombinant DNA technology, and the enzymes are restriction endonucleases, alkaline phosphatases, and DNA ligases. And in the next lesson we will discuss about the second tool of the recombinant DNA technology, and that is a vector. Thank you.